All right. So chapter 12 was about metabolism. And in fact, the entire semester has been building up to this because we've been learning all about what atoms are, how they bond together to make molecules, and then what the biological molecules look like that are involved in metabolism. So this chapter is kind of a summary almost of everything that we've learned this semester. So what we know now is that we have three major kinds of macronutrients that we consume. And those are the polysaccharides that we refer to as carbohydrates, the triglycerides that we often refer to as fats, and polypeptides that are the proteins that we eat. So these carbohydrates, again, are just polymers of sugar molecules or monosaccharides. Fats are a condensation of glycerol and fatty acids, and proteins are polymers of amino acids. So when I say these words, you want to try to get yourself to think about what the structures of those molecules look like. Those are our macronutrients. We eat those macronutrients, and what we learned in this chapter is that all of those macronutrients first get hydrolyzed into their smaller pieces, and then through different metabolic pathways, they are converted to a 3-carbon piece, which is pyruvate, and then or just directly to acetyl-CoA, which is a 2-carbon piece that enters the citric acid cycle and produces energy for our cells. The other fate of acetyl-CoA is to be stored as fat in the form of triglycerides, or to be stored in the form of a polysaccharide called glycogen. So those are the two main fates of our food: energy or storage. And then storage, of course, can be broken back down and be used as energy through the metabolic pathways that we already learned about when we talked about catabolism. So here's an overview of all metabolic pathways in your body that we talked about in chapter 12. And we focused on the blue arrows, which are catabolism, and some of the pink arrows are mentioned briefly, which are anabolism, or basically building back up. So the main gist of the catabolic pathways is that no matter what you eat, protein, polysaccharide, or lipid, the first thing that happens is they go to the digestive system. And in the digestive system, which includes the mouth, the stomach, and the intestines, they are broken down into their smaller pieces so that they can enter cells. So these blue arrows right on this line would represent hydrolysis reactions. Below the line would be inside of our cell in the cytoplasm. And so once these proteins are digested or hydrolyzed to amino acids, the polysaccharides are digested or hydrolyzed to monosaccharides, and lipids are digested or hydrolyzed to glycerol and fatty acids, then they are converted to pyruvate, so a 3-carbon piece. These molecules are broken down to a 3-carbon piece, which then enters the mitochondria, or in some cases, directly broken down to acetyl-CoA. So the ultimate fate here is to get everything broken down into smaller pieces, 3-carbon pieces, 2-carbon pieces that can then enter the citric acid cycle. And the only molecules that can kind of bypass the pyruvate acetyl-CoA are some amino acids that can enter the citric acid cycle directly after transamination. But what happens in here, like below this line, so this line here would represent the mitochondrial membrane. So all of this other metabolism is happening inside the mitochondria. So that's this organelle here. Most of it happens in the matrix of the mitochondria. So you could kind of write that this is the matrix here, not just general mitochondria. The citric acid cycle happens in the matrix, but the electron transport chain happens right on this inner mitochondrial membrane, this kind of wiggly membrane here. So here we've got inside the cell and then in the mitochondria. Now, what you'll notice is I'm going to make this chart smaller, and then I'm going to highlight individual places. And I'm going to start where we started in the chapter, with the catabolism of monosaccharides, and specifically glucose. So we use glucose as the kind of key example because that's the only sugar that can go through the entire glycolysis pathway. So glycolysis would be represented by this blue arrow here. So that's why I've highlighted it here on my little figure. So glycolysis is the catabolism of glucose. The outcome is you take one glucose molecule and you end up making two pyruvates. So overall, glucose is converted to 2 ATP, 2 NADH, and 2 pyruvate. So I'd like you to know some little details about this entire metabolic pathway. I don't expect you to memorize this, so please don't waste your energy memorizing this, but understand that glucose is phosphorylated, right? So there's an energy investment stage here. That's what these first 5 steps are. We phosphorylate glucose, we isomerize it into fructose, so we make it into an isomer. We phosphorylate it again, and then we break it apart into 2 molecules of glyceraldehyde 3-phosphates, which then can be converted into energy generation. So the glyceraldehyde 3-phosphate will produce an NADH molecule and then an ATP molecule through transferring of the phosphates. And eventually we end up at pyruvate after another generation of ATP. So if you notice this whole process here, it's only written out once, but it does occur twice because you started with 2 molecules of this glyceraldehyde 3-phosphate. So we'll have to double all of the output from the second phase, which is the energy generating phase. And the outcome is we put in 2 ATP above, we got out 4 ATP below, and an NADH, and then basically we have to subtract, right, and get the net output. And so the net output of this whole thing is 2 ATP, 2 NADH, and 2 pyruvate from each molecule of glucose. Once glucose is converted into pyruvate, then it's oxidized into acetyl-CoA and it loses a carbon in that process. So that's what this is. So notice I've highlighted a different place on my overview over here. And so this is the pyruvate oxidation step where we're converting pyruvate to acetyl-CoA. We kick out a carbon dioxide molecule. That carbon dioxide comes from right here. And the 2-carbon piece from pyruvate ends up on the coenzyme A molecule, and it's called acetyl-CoA. And in that process, we also produce NADH for each pyruvate. So for each pyruvate that goes through this oxidation, we get 1 acetyl-CoA, carbon dioxide, and an NADH. That carbon dioxide, it is exhaled because it's a waste product. And we talked about that this semester as well, how carbon dioxide is buffered in the blood with acid and then exhaled through the lungs. The acetyl-CoA will go through the citric acid cycle, and that's what's shown here. So the citric acid cycle is an energy generating cycle. The overview, or the net result of 1 acetyl-CoA going through the citric acid cycle is that we lose both of the 2 carbons. So each of the carbons that entered as the acetyl group will end up leaving its carbon dioxide. So they will be converted to waste. Every carbon that came from glucose ends up as carbon dioxide waste. We also get 3 NADH, an FADH2 molecule, and ATP. So the ATP that's directly generated from this is useful, but there's a lot more energy to get out. Before I talk about that, I just want to point out that what's on this slide shows you what happens with 1 pyruvate, but if we were talking about complete catabolism of a monosaccharide or glucose specifically, we would have 2 pyruvates to go through this process, and so you could double everything here. So you could double the amount of acetyl-CoA and NADH and carbon from this step, and then when you take that acetyl-CoA, those 2 acetyl-CoAs, and go through the citric acid cycle twice, then you would double this here. Now, the ATP is useful, but we also want to get more energy out of the NADH, FADH2s that we're getting from pyruvate oxidation and citric acid cycle. And I'm going to mention that in a minute. So the other kinds of molecules that we can use for energy: lipids and proteins. So let's look at lipids. So lipids is over here on our chart. We haven't really talked much about glycerol, so I'm going to focus only on the fatty acids. Fatty acids are used for fuel through beta oxidation. We talked about this just late last week, where you take a fatty acid, you invest 2 ATP to convert it to a coenzyme A. So there's the S-CoA molecule, or the coenzyme A, attached now to your fatty acid. And then each cycle of beta oxidation will cut this 2-carbon piece off, or the last 2-carbon piece essentially. You generate an FADH2 and an NADH molecule for every cycle of beta oxidation. And the number of cycles you need is 1 less than the number of carbons divided by 2. So once again, here we have a lot of these reduced dinucleotides being produced, the FADH2 and the NADH. There's actually no ATP actually directly generated from beta oxidation like there was in the citric acid cycle. So again, I'll talk about the fate of NADH and FADH2 in a moment. They're way down here, but we're gonna focus over here for 1 minute on the protein catabolism. So if your body's preferred fuel sources are sugars first and fats next. Proteins, not so much. Proteins are catabolized with kind of the intent of building more protein. But when your body does not have enough energy and there is not enough sugar or fat around, it will start breaking down proteins to generate energy. And it does that through transamination. So transamination is a process by which the amine group is removed from an amino acid. So when you remove that amine group, you end up with a keto acid. The ammonium here that is generated is toxic, and so it must be removed through the urea cycle, which basically converts it to urea and then is excreted in urine. These keto acids can be used for fuel. Depending on what family the amino acids are in, they will enter at different points in the cycle. It's not important to memorize that alanine is a 3-carbon family amino acid. It's just important to understand that if I say alanine is a 3-carbon family amino acid, where does it enter? If it's of a 3-carbon family, it enters at the only 3-carbon intermediate. If I tell you it's an amino acid from the 4-carbon family, it enters at one of the 3 different 4-carbon amino acids. And if it's of the 5-carbon family, it'll enter here at the only 5-carbon intermediate, and this 2-carbon family will enter here at acetyl-CoA. So this is not your preferred source of fuel, but it'll get you by in a pinch, basically. And the reason it's not your the preferred source of fuel is because it's not as rich in energy. So it's less bang for your buck. Amino acids take more work and get more energy out when you metabolize them, because they can't go all the way through the citric acid cycle unless they're up here. Many of them can't go all the way through the citric acid cycle, and then there's also no energy generated in their catabolism like there is in the catabolism of polysaccharides and lipids. Okay, so now in the catabolism of glucose and the catabolism of lipids, we've made a lot of these reduced dinucleotides. Those reduced dinucleotides then can be used as energy if they are oxidatively phosphorylated in the electron transport chain. So this is the sequence of proteins. So they're all touching each other in real life, and they're polypeptides, right? These are just cartoon versions of a protein, but all of these complexes 1, 2, 3, 4, and 5 are actually proteins that are all in a complex together. So they have some quaternary structure that's interesting because they are interacting with each other, but they are embedded in the membrane of the mitochondria, the inner membrane. They are membrane proteins. And basically, they catch electrons from the oxidation of NADH and FADH2, and those electrons are shuttled as if through a wire to complex 4, where the hydrogen ions and oxygen are converted into water. This is a very exergonic process. It's very energetically driven. So, you probably know that oxygen is flammable. Oxygen gas is flammable. Hydrogen gas is also flammable. We don't have oxygen and hydrogen sitting around in our atmosphere because when you mix oxygen and hydrogen, they are explosive the reaction is explosive to make water. This is a well-controlled reaction in your cells, but it is still very energetically favorable, very spontaneous. And so it draws electrons through and drives a process which phosphorylates ADP to ATP. So it provides the energy for this last complex to actually do the phosphorylation and store energy in the phosphoester bonds of ATP. The energy transfer molecules, their structures are here. So these are nucleotides or dinucleotides. So acetyl-CoA looks like that. There's just the little acetyl group that comes off in the low energy version. So we have high and low energy versions. And then here are our NADH, FADH2, and ATP. So acetyl-CoA will go through the citric acid cycle, that's what CAC means, to make NADH, FADH2, and ATP. And then any NADH and FADH2 that are produced in that citric acid cycle are then converted to ATP, or the energy in them are converted to ATP through the electron transport chain and oxidative phosphorylation. So that's the gist. That's the overview of metabolism. And one of the ways that you can test yourself to see if you understand all these parts is to fill out a blank worksheet like the one that I gave you in the activity. So if you haven't done this already, please do, and let's ask questions about it and work on it together if you haven't already. So there is a list of a bunch of different things on here that you should be able to label on this diagram. And then there's also things that are blank. And so just make sure that you can label, as many of the arrows or boxes on this chart as you possibly can. You should expect something similar to this on your final exam, maybe not exactly the same, but similar. So another really great way to study is to draw your own diagrams and make sure that you have filled in all the details from chapter 12. Then we also need to understand what each one means. So for example, if you know that polysaccharides to monosaccharides is a hydrolysis reaction, you also need to know how to draw that hydrolysis reaction just to kind of check all your boxes.